Aloha. Great, so we're on to symbiosis in soil organisms, okay? So symbiosis, right, is the phenomenon where two organisms grow more efficiently, produce more efficiently together than they do by themselves alone, right? 1 + 1 = 3 is symbiosis, okay? And our two largest phenomenon of symbiosis that we see, especially in the soil, right, is mycorrhizal and biological nitrogen fixation symbiosis, okay? So mycorrhizal is with fungi, and biological nitrogen fixation is generally occurring with bacteria, okay? Mycorrhizal associations are one of the most ecologically and economically important roles of soil fungi without a doubt. Hopefully you all have been somewhat familiar with symbiosis between mycorrhizal fungi and plants, but if not, this will be a pleasant walkthrough. And our BNF, biological nitrogen fixation, called the second most important biological reaction on the planet. As we'll look into, this is where we get a lot of our organic nitrogen, okay? Wish people were in this class so I could ask some quick trivia, but nobody is, right? So nitrogen is generally one of our most limiting nutrients in our ecosystems, in our croplands, in our forests, in our jungles, in our grasslands, okay? But, right, last we looked, nitrogen makes up a huge component into gas, right? It's a huge component of the atmosphere. And so how can that be that all of our ecosystems are mainly nitrogen limited when we have so much in the air? If we remember our chemistry classes, then we'll remember that N2 gas has a triple bond, right? Super, super strong triple bond holding those two nitrogen atoms together, keeping them in a gas form, okay? It takes a whole lot of energy to break that bond. And biological nitrogen fixation is a very acute way that organisms have developed a method to split, to take that huge energy required, to generate that huge energy required to split those into gas molecules. So the other way that N2 gas is delivered, right, to environments in general is from lightning, right? Actually, lightning deposition nitrogen, right, from when lightning strikes, a lot of the N2 gas that's floating around actually gets blasted apart by the lightning, and then those free nitrogens hug up with some hydrogens, right, and then get hug up with some oxygens and come down to the ground, right, and come down in the rain. There's acid rain, okay? But aside from lightning deposition, which in some places can be up to about 50 or so pounds of nitrogen per year, which is not nothing, okay? The majority of our nitrogen in our terrestrial systems, and marine systems too, is coming from biological nitrogen fixation. Okay, so mycorrhizae. The mechanism by which mycorrhizal associations really work is one of our themes of the class is surface area, okay? Mycorrhizae have a great effect on plant uptake of nutrients, right? Because they provide a lot of extra surface area connected up with the roots to then mine and deliver nutrients to the plant, okay? So we have a couple of different kinds of mycorrhizae, and we will get there for a second. You can buy mycorrhizae, right? Generally, it occurs semi-naturally depending on the type of environment you're in. But you can also buy products, right, to facilitate these symbiotic relationships in your own home or garden or potted plants or what have you. And again, the main mechanism by which these associations aid in plant growth, okay, is extension of the rhizosphere, right? Larger root systems, essentially, right? It's the mycorrhizal fungi and hyphae that is essentially tying into the root system and then mining nutrients for the plant. But it's the extension of that root system that ups the nutrient uptake of plants. And so we have a huge number of plants that form mycorrhizal associations. Like 90% of plant species on the earth do. So most of our angiosperm families, as in i.e. Regular old trees and plants, right? All gymnosperm families and many mosses and ferns and liverworts, even those more basic plants have, okay? Now we have our two main forms, which are ecto and endo, or our buscular mycorrhizae, okay? And our ectomycorrhizae are more common with our pines and conifers. About 10% of the total plant life, right, does these ectomycorrhizals. But then the more common one found in about 90% of plant species, right, is endo or buscular mycorrhizae. So, this association is a symbiotic one, okay? That means our energy pathways are going both ways, right? Our trees, for instance, if we're going to go with that example, our trees are really being helped by the mycorrhizae in that they are getting provided more nutrients from the more extended rhizosphere, right? What is the mycorrhizae getting? Mycorrhizae is getting what the tree is producing, which is sugar from photosynthesis, right? Carbohydrate. Plants trade carbohydrate for increased water and nutrient uptake. We see this effect on phosphorus here in many of our Hawaii soils that have very low phosphorus availability, right? We can see a large enhancement of that with mycorrhizal associations, okay? But the mycorrhizae is taking energy from whatever plant it's associating with, so you don't get something for nothing, right? So still some energy is being given to the mycorrhizae, which is not in the context of agronomy, right? It's not the crop that you're growing, okay? It's nothing for free. So, nice little graph showing phosphorus uptake in plants with our buscular mycorrhizae or not. So, the takeaway from this figure is what? Herman? What is it? Look at it! Look at it! Right? The takeaway from this graph is that shoot dry weight on our y-axis we can essentially think of as yield, okay? And look at this. We've essentially topped out in our yield with our buscular mycorrhizae, applying only 40, we can think about this as the amount of P added in terms of milligrams P per kilogram of soil. We can essentially think about it as pounds of phosphorus, right? So, excuse me, to look at this, with our buscular mycorrhizae, all we need to put on is 40 pounds of phosphorus, for instance, okay? But if we don't have mycorrhizae, in order to get those same numbers, we've got to put on 4 times the amount of nutrient, okay? Right? So these kinds of associations are super important, especially these days when we're starting to revise the old agronomic paradigm of nutrient application year after year, regardless of whatever's left over from the one before, right? And where economics are becoming a lot more tricky in a lot of farming practices, right, and production systems. So we're really looking at ways that we can cut down on our inputs, both for ecological and economic reasons, right? Who wants, I don't know how much it'll be to integrate some mycorrhizal associations in this situation, but I could probably guarantee it'll be cheaper than 4xing your phosphorus, right? So, in addition to helping out the plants get their nutrients and water, right? Mycorrhizal associations are really great when it comes to soil aggregation. We actually saw a number of times looking at, when we were talking about soil physical properties and aggregation, many times we saw a little in those little diagrams, a hyphae, little hyphae going through everything, mainly at the macro aggregate scale, right? So they're coming right back up again, front and center, right? That's exactly what they're doing. Those little hyphae that extend the root rhizosphere so much, right, are also actively growing and actively holding soil colloids and particles and other dead detritus together, right, and forming soil aggregates. So a question might be, what would be one method to enhance soil aggregation, right? What would that be after this context? Of course, it would be to introduce or enhance the mycorrhizal, the fungal organism populations in your soil, right? All right, biological nitrogen fixation. Biological nitrogen fixation is the association of bacteria with plants, right? Bacteria essentially set up shop in plant roots, form little microhabitats for themselves, and just the same way as mycorrhizal, in exchange for carbohydrate or sugar, the bacteria splits into gas, nitrogen gas, and provides mineralized nitrogen to the plant in exchange for carbohydrate. Okay, so we have a couple of different kinds: our obligate symbioses, which is both are necessary for the symbiosis, right? And our associative, which can or cannot need or don't need an association. Okay, and so our rhizobia is our largest bacteria in terms of biological nitrogen fixation. Largest not in size but in spread, right? In the areas in which we see them. Generally, these are hosted by all of our legumes, such as our soybeans, right? Our cowpea, etc. Great number, all of our clover, right? And then we have our cyanobacteria, and our associated symbiosis a lot of times is cyanobacterial bacteria and diazotrophs, and in association with higher plants. So the mechanism: these bacteria, such as Rhizobium, etc., have an enzyme called nitrogenase. Okay. NN is our N2 gas with that triple bond that we see, right? Takes 8 hydrogens and 8 electrons, so a lot of energy actually, right? And then we have our nitrogenase in the presence of iron or molybdenum, and then we're getting our 2 ammonias, right? 2 NH3s, okay? And an H2 set up. So, and here's our enzyme complex as is registered by Lancaster et al. In 2009, and they mapped out how this enzyme was structured in order to gain more insight on how it is able to split such a strong bond. This process, right, of biological nitrogen fixation, that's splitting the the N2 into ammonia, NH3, right? Does not like to be carried out in the presence of oxygen, okay? And so leg hemoglobin is, let me back up a little bit. These processes take place in the nodules, which are the little habitats that the rhizobium, right, set up shop in the roots of the plant, right? After an infection of the bacteria into the plant, a little nodule forms where they set up and reside, right? And so these nodules, within those nodules, a field test for these nodules is you can go grab a soybean plant, okay, and check for nodules. And then a healthy check would be to take your pocket knife and cut one of those nodules open and see if it's red, right? The red is the leg hemoglobin, which is similar to the hemoglobin in our blood, right? Which binds with oxygen and transports it actually for us. The leg hemoglobin in these root nodules binds really quick with oxygen as a means to exclude oxygen from the reaction center, okay? Leg hemoglobin was actually isolated and was a big breakthrough for the Impossible Burgers and Impossible Meat guys, because it was able to give it that red blood looking deal. And we also have heterocysts, which are cyanobacteria symbiotic biological nitrification. So the azola anabina complex that is in rice and kalo patties, right, utilizes this. This is a great method for essentially for free, right, integrate nitrogen into your system, right, of using an azola anabina complex that have nitrogenase capabilities, right, and as they grow and die off and grow and die off, there are dead bodies, right, that have fixed nitrogen from the N2 gas in the air, settle into the patty on the bottom, and then put that nitrogen into that system. Okay. So biological nitrogen fixation is used in a number of ways. Okay. A lot of our cover crops are biological nitrogen fixers, right? Our green manure are many of our sources of just organic matter, compost in general, right? We will look for a big component of that to be a biological nitrogen fixer in order to get that, essentially get that free nitrogen, right? Before the Haber-Bosch process in the 40s and 50s, and when it got industrialized in the 50s and on, right? This was our source of nitrogen, right? Was being sure to close our compost loop, right? And our production and our byproducts and our waste and our livestock production and their waste, right? We used to have to close those loops, keep those loops really tight, because we couldn't, if we lost some nitrogen from those systems, right? If we didn't take the cow manure and put it back on the cornfield to grow corn to feed the cow, right? Then our corn is going to start going down, and we're not going to be able to keep as many cattle, etc., right? And after the Haber-Bosch process, we no longer needed those two systems to be integrated, essentially, right? We just go buy nitrogen fertilizer from the store and not have to worry about keeping all those loud old animals just to put their poo on the field. Okay, let's call that a video and finish up with ecosystem engineers. Aloha.